Hello, I'm Alexandra Bowden, and I am the clinical lead for the Duchenne program at Audentes Therapeutics. And I'm excited to share today an overview of our exon skipping gene therapy approach. And additionally, we have Professor Flanagan from Nationwide Children's Hospital joining us to provide an update on the research that he and his team are pioneering using a gene therapy vector designed to induce exon skipping for exon 2 duplication-associated Duchenne muscular dystrophy. So before diving in, I'd like to take this opportunity to thank PPMD for the invitation to participate in and for hosting this conference to ensure that we all continue on this shared mission to explore and develop potential treatments for the Duchenne community. So this next slide is just a disclaimer slide to let you know that the work that we discuss today is with respect to the research of experimental or investigational products. So what and who are Audentes? Well, Audentes is made up of a group of individuals who are committed to developing genetic medicines that have the potential to improve the lives of patients. And the roots of our company are really reflected in our name, which is derived from the Latin verb audeo, meaning to be bold or to have courage. And it's these characteristics that we continue to observe in the patient communities that we hope to serve, that inspire us to be bold, to think outside the box, and to get stuff done at work every day. So how do we intend to do this? Well, we are exploring several investigational therapies based on an adeno-associated virus or AAV-mediated approaches to treat genetic diseases. And we currently aim to deliver these treatments with a one-time systemic administration of the AAV-based genetic medicine. We're exploring 3 different modalities: gene replacement, exon skipping, and RNA knockdown gene therapies. And within this presentation, we'll provide an overview and focus on the research that we're conducting for Duchenne muscular dystrophy, namely exon skipping gene therapy. Another capability of Audentes includes the internal manufacture of all our investigational products at our large-scale Good Manufacturing Practice, or GMP, facility in South San Francisco. And we recently announced the expansion of our manufacturing facilities to include a state-of-the-art gene therapy facility in North Carolina. Now, having our own manufacturing facility allows for our direct oversight of the quality and supply of these genetic medicines. Earlier this year, Astellas acquired Audentes to be the Center of Excellence for their newly created genetic regulation focus area within the broader Astellas organization. So this is just an overview of the Audentes pipeline. In addition to our first and flagship program, MTM1 gene replacement for X-linked myotubular myopathy, for which clinical trials were initiated back in 2017 and currently in a pivotal study phase, Audentes is investigating AAV-based genetic medicines to treat other rare neuromuscular conditions such as Pompe disease, which will also take a gene replacement therapy approach, myotonic dystrophy, exploring both exon skipping and vectorized RNA knockdown, and exon skipping gene therapy for Duchenne. Now, Audentes' initial programs for Duchenne will target duplications of exon 2, and you'll hear more about the ongoing research at Nationwide Children's Hospital shortly. And this product may also have the potential to treat mutations in exons 1 through 5. as well as exploring other treatments for mutations amenable to exon 51 skipping and exon 53 skipping. Now, all of the investigational approaches for Duchenne are in discovery or preclinical stages at this time, but these timelines are not to scale or relative to each other, and each of the products will progress as we learn more and more about the different candidates. So, as we know, the fundamental issue in Duchenne is caused by mutations in the DMD gene. And the DMD gene is the largest known human gene, made up of 79 exons. And these exons link together to form instructions for making the dystrophin protein. And in mutations where exons are missing or faulty exons are present, the instructions do not make sense anymore, and the gene is incapable of making enough or any of the dystrophin protein, which is critical for muscle health, strength, and function. So given the underlying cause of Duchenne, there are different approaches that are being explored to restore meaningful levels of functional dystrophin and preserve muscle integrity and function. So two of these approaches include exon skipping, which has been most extensively studied with naked antisense oligonucleotides, or ASOs, and the mini or microdystrophin gene transfer using AAVs. ASOs are small pieces of DNA that can mask or hide the exons to be skipped based on the specific mutation, so that the remaining exons can fit together, providing instructions to produce a shortened yet functional version of dystrophin. However, these naked ASOs have been hampered by poor distribution and cellular uptake, as well as rapid clearance, resulting in the need for high doses and frequent administrations. The other technology that's being studied is the microdystrophin gene transfer with AAV systemic delivery. And the AAV is used as a vector or a delivery vehicle because it can carry a new gene or genetic material into the appropriate cells of the body. Now, because of the large size of the dystrophin gene, researchers have had to think about reducing the size of the gene to fit within the packaging capacity of the AAV vector, which has led to the design and development of smaller versions, that is, the microdystrophin genes. So, by combining exon skipping and AAV technologies, namely exon skipping gene therapy, the weaknesses of one modality may be addressed by the strength of the other, with the potential to produce efficient and durable exon skipping, resulting in the synthesis of dystrophin in the tissues where it's naturally expressed. And using this strategy, AAV functions, again, as a delivery vehicle to introduce specific antisense sequences designed to skip over the faulty section or exons of the DMD gene, with that goal of producing meaningful levels of a shortened version, and for some mutations, a near full-length version of functional dystrophin for an extended period of time following a single administration. So with this approach, there is the potential of overcoming delivery, uptake, and clearance problems that may limit the earlier exon skipping approaches and result in a more complete version of the dystrophin protein. Now, as with all AAV-based gene therapies, it is not yet known how long the gene therapy will last, and it's important to note that the length of the dystrophin produced will be dependent upon the specific mutation. So outcomes associated with different types of dystrophin are being studied both across the clinical presentation associated with Becker muscular dystrophy and in clinical trials of microdystrophin gene transfer. And mutations in the DMD gene that maintain that intact or open reading frame, allowing for a semblance of the instructions to come together, produce shorter but functional dystrophin, and resulting in Becker muscular dystrophy, which is generally milder than DMD. The effect of even larger deletions, like those carried by the microdystrophin gene, are currently being investigated in ongoing clinical trials. So dystrophin restoration with exon skipping has the potential to produce the largest dystrophin protein possible, dependent on the mutation in muscles of individuals with DMD. And the dystrophin restored with exon skipping, again dependent on mutation, also has the capacity to retain functional domains of the dystrophin protein, including the N-terminal, portions of the central rod domain, the cysteine-rich domain, and C-terminal, each of which contribute to the structure, organization, and function of the protein, including the purported role of the C-terminal domain in the heart. So at this time, I'd like to welcome Professor Flanagan to review their innovative research with a gene therapy vector that's designed to induce exon skipping for exon 2 duplication-associated DMD, laying a cornerstone for this investigational technology.
Alexandra, thank you for the introduction to our program and for the great introduction to technologies that Audentes is taking for the treatment of Duchenne muscular dystrophy. I'm happy to share with you some of our work in exon 2 in just a few brief slides. We've actually presented much of the preliminary work at PPMD meetings in the past years. But to take this concept to the clinic, we've begun with duplications of exon 2. Exon 2 is the most common duplication mutation. It accounts for about 10% of all duplications, and nearly all patients appear as typical DMD, typical in severity of disease. In contrast to patients who carry a duplication of exon 2, we observed some years ago that patients who have a deletion of exon 2 have nearly no symptoms. And we showed that these boys make a dystrophin protein that begins within the coding region of exon 6 because of the presence there, presence just before there, of what's termed an internal ribosome entry site, or an IRES. An IRES can serve as a locus for the assembly of the ribosome, the translational machinery for a protein. So this is an example of alternate translational initiation. And what's very important, we know from patients who express this type of dystrophin that men who express it can walk into their 70s or even their 80s, as we first described when we described this form of dystrophin in a paper in 2009. Because of the presence of this IRES, exon 2 makes a great therapeutic target for exon skipping. If we skip only one copy of exon 2, we make a totally normal messenger RNA, which is the final blueprint for translation of the protein. And when that's translated, we'll get entirely normal full-length dystrophin. If we're successful enough to skip 2 copies of exon 2, we still make this highly functional IRES-driven dystrophin isoform. And the natural experiment of the men who carry this and express it and walk into their 7th or 8th decade tell us just how efficient it is. In order to test this, we made a mouse that carries a duplication of exon 2 called the Duke2 mouse. It mimics the features of the standard MDX mouse that many past experimental therapeutic development has been demonstrated in. And we generated a vector encapsulated within AAV9, and we call this a U7 ACCA vector. It contains the U7 small nuclear RNA, a non-coding gene that is tagged with a sequence that directs it to the splice acceptor site or the splice donor site of exon 2. And because we can fit 4 of these within one vector, that's how we created our initial vector. So we refer to it as U7 ACCA. We've demonstrated a lot of this data previously, but we'll show that, in fact, we showed proof of concept of the efficiency of this approach in a duplication 2 mouse model. This is a slightly confusing figure, but on the left side, what I'll show you is each of those bars represents an average amount of the different forms of messenger RNAs that we find in different muscles of animals. Each cohort here represents at least 5 animals. And looking on the left-hand side, you can see that with increasing doses, we see increasing amounts of blue or purple bars, and that represents the percentage of either wild type, that is 1 copy of exon 2, wild type messenger RNA, or the percentage of deletion 2, we represented by the symbol delta here, of delta 2 messenger RNA that contains no copies of exon 2. A reminder, each of these transcripts or messenger RNAs is therapeutic. What we expect from the light blue transcript is full-length dystrophin, and what we expect from the purple transcript is the iris-driven dystrophin that's missing a small fraction of the amino terminus of the protein. And on the right, you can see that we have robust expression of dystrophin in animals who are treated with this. Here, animals treated at about 8e to the 13th vector genomes per kilogram. We have significant expression in skeletal muscle, here in tibialis anterior, in diaphragm and heart. What you see on the left column is animals, wild-type animals, not the Duke2 animals, but wild-type expression. In the central column is untreated Duke2 animals. And in the right column, these tissues treated with this vector. So we know that we get robust expression of a very highly functional dystrophin protein with this. This preclinical data supported our ongoing clinical trial, first in human clinical trial of systemic delivery of this AAV9 U7 ACCA construct for patients with duplications of exon 2. The trial is sponsored by Nationwide Children's Hospital in collaboration with Audentes, and the principal investigator is my colleague, Dr. Megan Waldrop. Recruiting is by invitation only, and inclusion criteria includes having a confirmed duplication in exon 2, an age greater than 6 months and less than 14 years, and subjects must be ambulatory and currently taking steroids. I've included here at the bottom of the slide the reference number for clinicaltrials.gov for your use. To date, 2 patients have been dosed with 3 times 10 to the 13th vector genomes per kilogram. This trial primarily assesses safety, but the primary measure of efficacy is the change from baseline in dystrophin expression, as measured in comparison between the baseline and 3 months post-injection biopsies. In our first patient, the month 3 biopsy was delayed due to COVID regulations, but the blinded analysis is now underway. This first-in-human U7 small nuclear RNA trial may facilitate future U7-based exon skipping therapies, such as the AT702 product under development at Audentes. With that, I'll return the presentation to Dr. Bowden.
So thank you so much, Professor Flanagan, for sharing these insights. And of course, we look forward to learning more. And obviously, we're thrilled to be collaborating with NCH in their ongoing research. And before wrapping up, I'd like to take a moment to describe how Audentes plans to leverage this technology to create an efficient DMD development program. So Audentes' goal is to develop a platform approach. And this platform approach is predicated on the use of identical vectors that differ only in the specific antisense sequences tailored for each mutation amenable to exon skipping. And to that end, we are in the midst of optimizing AT702, AT751, and AT753 for their respective mutation-specific targets. And these are currently being evaluated in preclinical studies. So by leveraging this technology, it's possible that more than half of the DMD population could be addressed by just 8 exon skipping products. And a platform approach by building upon the manufacturing and preclinical knowledge that's gained across these identical vectors has the potential to enable an accelerated pathway for our development program. And I'd just like to point out that the initial programs are targeting mutations amenable to exon skipping that are bolded within this pie chart. So in closing, I'd like to thank Professor Flanagan and the team at NCH again for their pioneering research, and to PPMD for providing this opportunity to share Audentes' approach as a potential future treatment option for Duchenne. At Audentes, we really are committed to integrating the patient and family perspective into our work so that we develop clinical trials and treatments that are meaningful to patients and families, and where your input truly makes a difference. If you have any questions after watching this video or would like to contact our patient advocacy and engagement team at Audentes, please email patientadvocacy@audentestx.com. And we're already looking forward to participating in the live gene therapy panel on Friday, the 24th of July. And until then, thank you again.